A reminder that the maize meeting registration for early 2024 is now open. So you can already register and start making plans for the maize meeting. And I think without further ado, Sheng Kai, you want to take it away?
Yes, sure. So, hi everyone. So today I'm going to tell you about something about the nitrogen cycle around the root systems of different grass species. So why do I study nitrogen cycle? That's because we have a big nitrogen problem in modern maize agriculture. And in the soil, are three major forms of nitrogen: ammonium, nitrate, and organic nitrogen. And they're interchangeable via different microbial and chemical processes. But in an agricultural system, to boost yield, we spend a lot of money and energy to produce inorganic nitrogen fertilizers and apply them to the soil. And indeed, the application of fertilizer made possible the Green Revolution. However, it also has a big impact on our environments. So our crops are not able to uptake and use all the applied nitrogen. Particularly, the high mobility of nitrates makes it easy to exit the soil system, and this is either through gaseous emission of nitrous oxide or leaching with water. Either way pollutes our living environment significantly. Nitrous oxide has strong greenhouse effects, and nitrate runoff contaminates water resources. So a striking instance here is that the nitrogen runoff from US agricultural system has led to a tremendous dead zone in the Gulf of Mexico, as you can see here in the picture, which is already the size of New Jersey states. So because of this, we care about nitrogen cycle in agricultural system, and people have started thinking about ways to mitigate the problem. And one solution focuses on the soil nitrification processes. So by slowing down the conversion of ammonium to nitrate, we may be able to reduce nitrogen loss from the system. So recently, researchers have discovered that some compounds in the maize roots can inhibit the growth of nitrifying bacteria that catalyze soil nitrification process. And this trait is called biological nitrification inhibition, BNI. And those secondary metabolites with BNI activity is called BNI compounds. And this trait is not only seen in maize, but also in several other cereal or forage crops. They all produce all sorts of different compounds that can convey some BNI activities. So BNI seems to be a great breeding target for maize if there's room for selection. But I wonder, how far can we push BNI to? So we may want to slow down the nitrification process to a certain level, but we wouldn't want to stop the nitrogen cycle completely. And I believe that too much modification of the soil nitrification rates may also start limiting the nitrogen uptake of the plants if there's a preferred ratio between ammonium and nitrates. So I think we can actually get some hints by looking at the evolution of these traits in other species. And indeed, as we look across the globe, there's huge variation in the distribution and abundance of nitrates and ammonium. And the adaptation to these different environments did result in different ways for the plants to cope with the soil nitrogen. And the evolution of BNI activity may be part of the adaptive processes. So understanding whether or not BNI evolve in different grass species and to which extent the inhibitory effects could be will enable us to make more reasonable strategy to breed BNI activity in maize. And this is the major goal of this study. Or more generally, what does the rhizosphere nitrogen cycle look like in diverse grass species if BNI is not commonly seen at all? So we hope the evolution of wild and perennial grasses will teach us how to make the agricultural soil system more sustainable. Okay, so the best materials to answer these questions are in the Andropogonia tribe, which maize and sorghum are part of and are closely related to. So the previous efforts in the NSF PNM projects have been really helpful for the development of this study. And as introduced by Toby and Matt a few weeks ago in this webinar series, this project has made available lots of valuable genetic resources for this Androbogonian tribe. And as part of this project, Taylor Option Elder in Toby Kellogg's groups collected many live plant clones within this tribe. And with Nick's Apex cares, these materials live happily in our greenhouse at Cornell University, so that I can easily make some splits and perform a common garden experiments with them. So I've been working on around 40 different Androbogonian species, and most of these species are perennial grasses, except for Telepogon elegans, which is a wild annuals. And in addition, I also included maize and sorghum in the experiments as annual crop check. So plantlets and seedlings of this species are grown in the US Corn Belt soil in a common garden greenhouse setup. And here I'd like to thank Angela Kent and her graduate student Sarah Racklin at the University of Illinois Urbana-Champaign for sending us some field soil and their generosity on sharing valuable suggestions when we are designing experiments. And after 4 weeks of regular greenhouse establishment from the transplantation, I applied once ammonium to all these different species and then stopped fertilizing. And the same amount of water was provided after the fertilization of ammonium to all plants every day to control for the leaching. Then I collected multiple samples of soil pore water during the experiments, and at the end of the experiments, which is 4 weeks after fertilization, I collected samples of rhizosphere soil and iron exchanging resins pre-installed beneath the potting system. So these samples are for nitrogen traits measurements. And in addition to this, roots and leaf tissues from these different species were also sampled for RNA-seq profiling. So our nitrogen phenotyping is done by Brian Emmett's group at Iowa State, and his high throughput protocols made assaying hundreds of samples possible and efficient. So for all these different samples from different species, we measure the potential nitrification rates of their rhizosphere soil relative to the bulk soil without plants, the nitrate content in the soil, and the leached nitrate captured by the ion exchanging resins. So here are the distribution of these different traits. Importantly, all 3 traits show significant variation among different species. So the variance explained by the species range from 30% to 85% for different traits. And interestingly, we observed a striking contrast between the annuals and the perennial species in the traits distribution. All annual species, no matter domesticated or wild, they show a significant nitrification inhibitory effect, produce the least and lost the least nitrates compared to other species. And in contrast, most of the perennial species do not have an impact on their soil nitrification rates, or even in some species, there's a significant nitrification stimulating effect. So visualization of this trait variation on the species phylogeny indicates independent evolution of nitrification inhibition or nitrification stimulation in different lineages. So from these trees, you may also notice that the soil nitrification rates correlates quite well with soil nitrate content, as we would expect. However, soil potential nitrification rates does not correlate with the nitrate leaching. This implies some variations in nitrate uptake among different species. So we'd like to look further into this. So now focusing on the 2 uncorrelated traits, soil nitrification rates and nitrate leaching, we're able to classify the species into multiple groups with distinct rhizosphere nitrogen managing strategies. So at the moment, I call them the conservationists, the leachers, and the nitrate utilizers. These are just the names that I came up with for the talk today, but I'm happy to hear suggestions on how to better describe these different strategies from you all. So first group of species, the conservationists, includes all annual species and only one cultivated perennial species, vetiver. They inhibit the nitrification and thus conserve nitrogen in the system as ammonium. That's why I call them the conservationists, and I suspect that they may also evolve the preference for ammonium uptake in order to utilize the conserved ammonium in the soil. For the second group of species, the lichens, the nitrifying bacteria somehow benefits from their presence and thus produce lots of nitrate in the soil. However, the lichens are not able to uptake all of them, so unfortunately, most of the converted nitrates got lost from the system. In contrast, although the nitrate utilizer also stimulates the nitrification process, little of the nitrate is lost. So this suggests to me that they are actually good uptaker or utilizers of the nitrates. So now that we've seen such pronounced phylogenetic variation in the rhizosphere nitrogen cycle, we'd like to look further for the underlying genes. So what are the potential genetic changes that can affect trait variation? This includes genes presence and absence, pseudogenization, gene copy number expansion or contraction, and the regulation of gene expression. So we can test the impact of different genetic changes by a linear association model among the assayed species, where the Y is the trait of interest and the X stands for different genetic factors. But how can we measure these different X's? With the genome assemblies and RNA-seq profiling of these different species, we should be able to get some idea. So first, I identified the closest homologous gene groups across different species as the units of test. And this is done by a mini-prot search of the outgroup proteome against each of the Androbogonian genomes that are run in Hoover's lab and Amy here in the Buckler lab assembled. And Paspalum vaginatum is the closest outgroup of the Androbogonia that we can find. So with the best matching homologs for each of the ancestor genes, I then aligned the coding sequence of different species and estimated the genetic distance from the outgroup copies for each of the homologous genes in each species to represent their rates of evolution. On the other hand, I mapped the RNA-seq reads from each species to the homologous genes in each of the species to estimate the gene expression and dosage. So with the distribution of genetic distance to the outgroup, we can approximate the process of gene loss and pseudogenization with a binary factor x1. When the gene is absent or when it accumulates too many mutations, which leads to longer distance to the outgroup copy, I consider them lost or pseudogenized in that species and then assign x1 to 0. Otherwise, the homolog should encode a potentially functional protein, and hence x1 should equals to 1. On the other hand, the estimated gene expression is a continuous variable that represents the abundance of gene products, either via more copies or expression regulation. So we can model this impact of the variable directly in a linear model. So now we have two association models. One tests for the impact of gene loss and pseudogenization. The other tests for the effects of gene expression or dosage of a gene. However, we unfortunately have a very limited sample size in the testing framework here. So to avoid power issue, I chose to focus on a list of candidate genes only rather than all genes across the genome. So digging through the literatures, I put together a list of around 300 genes that includes previous GWAS hits for these traits, genes involved in the biosynthesis of known BNI compounds, and multiple families of transporters whose function may be relevant. So here's the results of the sequence-based model for gene loss and pseudogenization. What you're seeing here in these plots are the effect sizes of the functional gene copy presence on the x-axis and model significance on the y-axis here. So each dot here represents a gene. So among all tested genes, the functions of a PTR nitrogen transporter appears to be the most important for both nitrification inhibition and the prevention of nitrate leaching. Interestingly, the expression-based model also identified more transporters with significant effects on these nitrogen traits. This includes multiple nitrate transporter, ammonium transporter, and several ABC type transporter. Or their expression difference among species co-vary with the rhizosphere nitrogen traits. So there's quite a strong enrichment for the transporter in the association results compared to all other candidate genes. So this highlights that the functions and the expression of transporter genes, particularly nitrogen transporter, are more important to the rhizosphere nitrogen cycle compared to the genes involved in the biosynthesis of the secondary metabolites with BNI activities. So we would like to look into the expression and the functions of some significant transporters more closely in the diverse groups of species with distinct rhizosphere nitrogen traits. And as we hypothesized in the conservationists, the ammonium transporter are expressed at higher level compared to all other species. This makes totally sense because they inhibit the nitrification process, so ammonium is the major source in the soil for them to uptake. But quite interestingly, all the conservationist species have also a functional copy of the PTR transporters that we identify too, although there's not much nitrate in the rhizosphere for them to uptake. On the other hand, in the leacher species, the ammonium transporter expression is relatively low. Also, we can only find multifunctional copy of the nitrate transporter in this group of species. This explains their leaching problem because they simply don't have the ability to uptake much nitrogen. And in contrast, though stimulating the nitrification process too, the nitrate utilizers have a functional nitrate transporter to uptake the produced nitrate, and this prevents them from being the leachers. So we've seen the distinct rhizosphere nitrogen managing strategy in different species and the potential impact of some interesting nitrogen transporters. Next, let's see what shapes the observed genetics and phenotypic variations. So to answer these questions, we need to understand what the habitat environment looks like for these different diverse grass species. And Germano, another postdoc in our lab, has developed a neat approach to describe the habitat environments of different species with a principal component analysis. So different principal component in his analysis summarize the variation of different environmental features among the species range. For instance, the first and the second components from this analysis capture respectively the variation of temperature and precipitation related features in the habitats of different species. So by associating the environmental pieces to the observed traits in the focal species here, we should be able to infer something about whether the traits variation can be explained by the adaptation to the environmental factors varying on a specific environmental axis. So as a result, we can identify several environmental pieces that significantly co-vary with the rhizosphere traits. And very interestingly, these significant environmental pieces capture the variation of different soil characteristics in the original habitats of these different species. And this includes soil particle size, soil acidity, soil nutrient content, and the soil temperature stability. So when we look closer at this significant soil characteristics in the habitats ranges of the three groups of species with distinct nitrogen managing pattern in their roots, we can see that first, the leacher originally live on the soil with larger particle size, probably with more leaching problem already. And second, both leacher and nitrate utilizers live on more acidic soil. Third, the conservationists live on relatively richer soil compared to the other two groups. Furthermore, there are also some other climatic features like the interplay between rainfall and temperature, as well as the temperature stability of the soil may also play some roles behind the trait variation. So looking at this summary table, I hypothesized that the variation of this soil and climatic features may determine the microbial growth and composition in the habitat soils. And thus, this impose differential selection pressures on the locally adapted grass species, which results in the observed variation in the rhizosphere nitrogen cycle here. And it is important to highlight that the growth of nitrifying bacteria is actually limited when the soil pH is below 6. So this suggests that only the annual conservationists have experience or have the chance to experience the competition with nitrifying bacteria in the past. So it is possible that the inhibitory traits is actually an adaptive response to it. While the other species here, all these perennial species living on the acidic soil may not know at all how to cope with all the nitrifying bacteria in the maize field. So the bacteria just all compete them completely in our experiments. All right, so to sum up what I covered in the talk today, the first and the most important take-home message is that biological nitrification inhibition, BNI trait, is actually a convergent trait shared by all acid annual grass species, no matter they're domesticated or not. And the BNI activity in maize is already at the higher ends of the spectrum along the Androbogonian phylogeny. So there might not be too much space to push further the BNI activity in maize during their active growing season. Then, how are we going to solve the nitrogen problem in maize? When we think more about the nitrogen problem, we realize that there may be a timing issue. So now that we know the maize is an annual crops that already have a reasonably great strategy to limit their nitrogen loss from the system during their active growing season, but they cannot grow at all times of the year. And actually, most of the nitrogen lost in the maize agricultural system happens early spring, when the microbial communities start gaining activity, but the maize is still small or not even grown. Hence, we start thinking, if we can maintain the high BNI activity of the active growing maize off growing season or even over winter, we may be able to mitigate the problem more efficiently. And Sam here in Grore's lab here at Cornell University has been interested in this topic too, and he has started looking into the BNI activity of maize stoffers of different genotypes. So it will be really interesting to see what he will find out with the experiments. And in addition, our results highlight the importance of transporters as well. So I'm also thinking whether we can do something with the transporters to improve the sustainability of maize agriculture. So first, although the ammonium transporters are highly expressed in the maize, in this study, we're actually not sure about whether the observed high expression is an induced response to the high ammonium level specific in our experiments, or it is a real constitutive higher expression in maize compared to other species, even in other conditions. So if we can make the expression constitutively high for this ammonium transporter, this may make the maize even more competitive for ammonium uptake than the bacteria. Second, although there's a functional copies of the PTR nitrogen transporter identified in this study, it is actually lowly expressed in maize, so the expression is very low. So similarly, if we can upregulate these genes, it may also help the maize to take up all the nitrates more efficiently when they are produced, and thus further prevents nitrogen loss. So I'm really curious about the effects on the yield and the environmental impact when these genes, these nitrogen transporter genes, are overexpressed. So as you might have already noticed, I'm not a plant physiologist, so I may have overlooked many publications on these nitrogen transporters, but I believe they exist, and given their importance for nitrogen use efficiency too. So if you have any papers in mind, I'd be happy to learn more about this. And last but not least, exporter for BNI compounds will also be a good target for selection. But more studies are still needed for this aspect because we actually have little idea about the exudation of this secondary metabolites and the transporter responsible for them. So with that, I'd like to thank all the Buckler Lab members for their support, groups in UIUC and CMIT for their valuable inputs during the early phase of this project, Harvard staff for all the genome resources, and all of you for your attention. Okay, so do you have any questions?
Thanks, Sheng Kai. Folks can put questions in the chat or raise your hand. I wanted to start off, I had, I guess, two questions. On your correlation between leaching and nitrification, it looked to me like it actually might be a positive correlation, except for a couple outliers. And I wondered what your thoughts are and what are those outliers?
Okay, so you're talking about these pictures, these plots here, and yeah, I see your points on the outliers here, but I think the outliers
I was
here
at the top.
Yeah. Oh, this one on the top here.
That one and the one above 30.
Yeah, these are the only two species that lose more nitrogen compared to the bulk soil bank. I haven't tried doing the correlation, removing the dots, but I can give that a try. But I'm actually thinking of the lack of correlation might actually come with the dots here in this quadrant, and I think they're the interesting species that, in my opinion, because they produce a lot of nitrates by stimulating the nitrification process, but they don't actually lose much nitrates compared to the annual conservationists here.
And what are those two species at the top that you were mentioning?
Sorry, I don't remember exactly what the species names here, but I can double check. And yeah, maybe they are just not growing very well in a greenhouse condition, and that's why it's been an outgroups here and an outliers.
Yeah, I was wondering what the biological explanation was for that. That makes sense.
Yeah.
And in your distance plot from the alignments, it looks like you had multiple modes of distance. And yes. I thought I was confused by that.
Okay, so this plot here. So on the x-axis here is the genetic distance to the outgroup copy, and this is the frequency of it. Yeah, and yes, we have like one mode here and another mode here and yet another here. So yeah, I didn't explain this very well. The mode at 1 here is actually arbitrary assigned. I'm just assigning all the missing alignments as a distance of 1.
Sure.
So this is actually the distance of the gene. And then there's a second groups here that I am interpreting this gene copy as pseudogenized gene copy because they accumulate much more mutations compared to all other species. And
is there any way, I mean, do you know those are non-functional mutations, or is there any way that could just be subfunctionalization?
That could be possible. I haven't checked for yeah, I can actually do some calculation like DNDS to check if all those are non-synonymous or functional. Yeah, I can double check this.
Or just to look for mutations that actually interrupt the CDS, right?
Yeah.
There's a question in the chat from Jin Lang on, for these grasses, have you checked or measured their photosynthetic NUE? I'm wondering if they're BNI related with their productivity.
Actually, we haven't, because we were thinking about measuring these traits in the very beginning, but it's quite challenging, because if you look at the picture, I'm not sure if it's clear, but the leaf morphology of these different species varies a lot, and some traditional measurements for photosynthesis, like the based on the spectrometer, may not work very well. We test some, and it doesn't work very well on this trait, on this species. And calibration between species is also another problem, another challenge that we are facing here. But thanks for the question. I believe that, yeah, it could be relevant, but at the moment we are not able to answer.
And then you had the neat plot about when BNI is effective in an annual and when ammonia production is happening, and suggesting that maybe BNI and Stover could be a solution. But that implies that the real question you're interested in, sorry, nitrate, not ammonia, that isn't the real challenge that you're trying to face is annual nitrate production, and in which case would something that's maybe not quite as conservative as maize, but is a perennial and has some lower level of BNI throughout the entire year actually be a better approach?
So yeah, then let me see if I understand you correctly. So you're saying that if we have a perennial species and that is growing all year long, it will also solve the problem of nitrate leaching. Is this what?
Yeah, right.
Yeah, this I agree, but I do think a lot of the perennial species, they grow year long at their habitats. And there's also evidence, if I remember correctly, showing that perennial forage field is actually losing or have less nitrate leaching issue. And that's because of their year-long occupancy on the field. But in the case of annual maize, then yeah, we need to rely on either shifting the growing season or the Stover BNI activity. Or yeah, if we can do perennial maize, that may also solve the problem. But perennial maize may also have a strong hit on the yield, I guess.
Yeah. Thank you.
Thank you.
Let's see. Question from Ellie in the chat. Maybe you've already seen this, but here's a paper on Arabidopsis end use with a curated list of 300 nitrogen-related genes, including transporters.
Oh, let me see. Thanks for sharing this. I'm not reading a lot about the nitrogen transporter yet, so thanks.
Okay, if there are no other questions, you grab that link.
I have a question, a couple of questions. Hi, Sheng Kai. About the methods, when I usually see this association between traits and phylogeny, they usually correct by the activity by the genetic distance between the relatives. So in a GWAS, you would correct for the population structure with a kinship matrix. In these cases, you don't have to correct for the length of the branches.
Yes.
So that would be the expected neutral evolution between the branches. It depends on the length of the branches.
Yeah, thanks for the questions. I'm actually expecting that. So indeed, we need to correct for the species relatedness in this association model. So the real model should look like that, with the K here stands for the relatedness between species. And we did try an approach like a phylogenetics mixed model approach proposed by Michael Lynch several decades ago by controlling for the shared branch lengths between different species. But testing this approach on our phylogeny or our materials, we actually see very little structure among the species we have here. So our conclusion is that for this uniformly sampled species across the phylogeny, the relatedness may not have a strong impact on the association analysis. Yeah.
Yes. And I have a question about, can you show the distribution of your species again?
Sorry?
The distribution of the species, but they should be global, probably. The distribution of the species.
Oh, the distribution.
The geographic distribution.
Oh, the geographic distribution. I don't have the figure here, but
because I wondered. Yes, I see what you have there is like a bias towards temperate, but I'm not sure if that is the case.
Yeah, sorry. So this plot here, the species habitat records I'm showing here is actually a temperate, an example from a temperate grass species. So it's high bias to, the temperate area of America. But in his analysis, in Germano's analysis here, he goes into the database and then trying to download all the species habitat records of the different species and then summarize the environments of these different species in the metrics like this. Showing here.
Okay, because I have a trouble working with corn and phosphorus, because the measurements of phosphorus availability are very low. Well, they are bad in general for the tropics because there are lack of sampling among other things. So they are those are better. There are further information on the temperate zones. So yes, I was wondering if maybe the thing that you have correlated between phosphorus and your ambient might be correlated is with other things, because that was on the only thing. Thank you.
Thank you.
Okay. Well, thank you, Sheng Kai. Thanks all, and we'll see folks next week.
Thank you.